Good evening. I'm Yvonne Stepp for Science for the Public, and I welcome you to Contemporary Science Issues and Innovations. Tonight's discussion is about biological cells, specifically about their components called organelles that regulate cell functions, rather like the various organs in our bodies. More specifically, we discuss an issue of scale. What restricts the size of various organelles so that they can function inside a typically microscopic space? Our expert is Jané Kondev, the William R. Kenan Jr. Professor of Physics and HHMI Professor at Brandeis University. Dr. Kondev is a theoretical physicist who works primarily on problems of molecular and cell biology. You can see more information about Dr. Kondev in his lab on our event page on the Science for the Public website for tonight. So we'll learn how biophysics probes one of the most interesting enigmas of the cell. And as an additional treat, Dr. Kondev has agreed to describe an innovative program for teaching undergraduate students at Brandeis University. We're very pleased to welcome Jané Kondev. Welcome.
Thank you.
And I'll ask you to start us out. Take us back to the cell in general. So something about cells, and then to take us inside the cell for the organelles.
Yeah. No, the cell is an amazing object. So it's the fundamental unit of life, and unlike what is often thought of, it's not a bag of chemicals. In fact, it's intricately organized, and part of this organization inside the cell are organelles. These are objects that are somewhat smaller than the cell, which perform specific functions. Not unlike the way that functions of a city are separate, right? So we have power plants that produce all the electricity that we use, and similarly a cell will have an organelle called the mitochondria, which produces most of the energy that the cell needs in order to perform its functions. Then there's the nucleus, which contains all the DNA, and you can think of the nucleus as the place where information is stored and processed. There's things like, as we see in this picture here of a plant cell, there's something like the endoplasmatic reticulum, which is right on top of the nucleus, and that's where all the protein production and packaging is performed. So that's sort of like when we order our books from Amazon, the Amazon warehouse of the cell is this endoplasmatic reticulum. And what's amazing about these structures is that they have to be proportionally sized to the size of the cell. So even though they're made of tiny molecules, microscopic components, which are much, much, much thousands of times smaller than the size of the cell itself, these have to somehow self-organize into these much larger structures in a way that is very specific, that reaches a very specific size. And how this happens inside a cell is really one of the great mysteries of cell biology.
And just on that, before you go on in more detail there, do they move around? And are they in little packets themselves? Do they have to move around? Is that one of the issues for them for space?
Well, the organelles themselves, they won't be moving around much, at least not in the very dense environment of the cell. That's certainly true of the nucleus, but actually, if you think about like the mitochondrion, which produces, like I said, energy, sometimes those organelles actually have to be transported to very distant parts of the cell. For example, nerve cells can have these very long protrusions, which in some animals can be even meters long. And then you have to get these mitochondria, namely the parts of the cell, the organelles that produce energy, you have to get them all the way to the tip of this extension of the cell. And that does require movement. And actually how this transport of these organelles is orchestrated in time is also another fascinating problem.
It's just, there are a lot of things to consider. You were focusing, I think, a bit on the actual space in there. So they come in different sizes. These things are different sizes themselves. So everything is microscopic, but they're in different sizes, and they got to pack together in there. In your mind, are there any sort of underlying, rules for the size?
So that's really kind of one of the central questions that animates the research that we do in my lab, but also in many other labs around the world, where people are interested in this question. What are the fundamental rules that determine the size of these organelles? One of the reasons we'd like to know is because, you see, on one hand, we've decoded the genome. We know how to read the DNA code, and we know what the instructions are. And those instructions that are in the DNA code, they talk about the parts. They talk about the molecules that the cell needs to make in order to produce its function. But then comes the question, how do these molecules self-assemble into these structures that we see on this graph? And it's not at all clear where that information is encoded. And so we don't understand that code. And it's when that code gets misread is often that cells stop functioning the way they should normally, and that could lead to disease. Also, if we could manipulate that code, then maybe we could start making the cells that do our own bidding, which is kind of one of the things that's become very active in the field of synthetic biology, where people are trying to produce synthetic cells and organisms that perform useful functions, like maybe produce medicines or molecules that could be used in the context of energy, energy that humans need to do their own thing. Yes.
Right. So they can go wrong. They can go wrong. So that's one issue. And the other is how they are organized. This space thing appears from what you've said to be very important. Can you give us an idea of the space? They're not packed in there, one on top of the other.
Yeah, well, they almost are. So for example, inside a bacterial cell, which is a smaller version of the cell we're seeing here, one of the key components, one of the key machines that's inside that cell is something called a ribosome. The ribosome is the machine that actually makes proteins, and proteins are the Lego blocks of the cell. That's the building material of the cell. And these machines that make proteins, their spacing is about the size of the machines themselves. So that's like being in a crowded New York subway, right? Where essentially the distance between you and your nearest neighboring fellow passenger is roughly the same size as you know, the width of a human. So that's that kind of dense packing is typical of the cell. And even though we haven't spent in my lab as much time thinking about that, this is something that's been a real puzzle as well in cell biology for decades, which is how does all this hustle and bustle inside the cell that happens, all this transport and molecules moving around and organelles moving around, all because they need to perform their function. How does this all happen in this very, very crowded environment? And that's another open question, which we don't have answers to.
Are they communicating with each other? Is there some cross, so that distance and the way they're arranged might matter?
Oh, absolutely. That's a fantastic question. So for example, inside the nucleus, what's going on is the information that's encoded in the DNA is being read by very specific machines, and that information it's transcribed into molecules called RNA. But now these molecules need to be exported outside of the nucleus and captured by the endoplasmatic reticulum. And so the fact that because it's on the endoplasmatic reticulum, which is this organelle that abuts the nucleus, it's on that organelle that the process of translation of this RNA into protein happens. So there's this communication that happens between the two organelles, the nucleus on one side and this endoplasmatic reticulum on the other side, that is definitely sort of arranged in space by its mutual arrangement. So the fact that they're close by affects how fast and with what fidelity do these signals travel from one organelle to the other.
Two things. First of all, that space seems very important. If that space is off, the communication might go wrong. The other is the communication like physical or chemical or what?
Yeah. Well, it's chemical and it's physical. So I would say, I mean, this is one of the amazing, fascinating things that happens when you start looking inside the cell. You quickly realize that these boundaries that we've erected between the different scientific disciplines go away. Where physics starts and chemistry ends is not at all clear. So for something like an RNA molecule, which we just were discussing, it undergoes a physical process of diffusion, something that was, that's been known to the physics community since the 19th century, and essentially was crucial to our discovery of atoms in the first place. That process is present, and it's important for the transport. But at the same time, there's chemical compounds on these molecules that are used in order to recognize which molecule goes where. So there's kind of a sorting process. There's addresses, if you like, zip codes associated with these molecules, which are chemical, but then the process itself of transport could be physical. So all these things come into play when you're trying to understand how molecules sort of get from one side of the cell to the other in these various processes. Yeah.
This is probably naive, but it seems to be that sometimes chemists are over here and physicists are over here, and they might not be aware of what the other side knows. Is that possible? Did it take a while? I realize there's convergence now, but people were so reductionist before that, but the chemists and physicists both understand what's going on there.
Yeah, so it seems, yes, I mean, there's definitely...
they need to understand.
There needs, this convergence of the different science disciplines is absolutely important if we want to get at a quantitative understanding of the cell. That's something that's sort of been permeating the sciences recently, but it's not a very recent invention. I mean, there have been sort of looking through the history of biology, for example, there have been many instances in the past where very fertile communication between the physicists, the chemists, and the biologists led to important discoveries. I mean, Francis Crick was a physicist of the Watson-Crick fame, and so on and so forth. So a lot of, I think, kind of, I would say a lot of the major breakthroughs in biology have often happened, not always, but have often happened when scientists from a variety of disciplines have come together to look at a fundamental biological puzzle and then have applied their different disciplinary tools in order to address the puzzle.
Right, and here you really need it.
So here I think we're definitely seeing that convergence happening again. People are coming in with all the tools from cell biology, from all the tools molecular biology, but then also physics, chemistry, and mathematics, computer science, all in trying to understand what is, the central question being, what is this code? What is this code that tells the cell how to organize its functions? And when I say functions, physically that means these organelles and the molecules that are associated with them, how to organize them in space and time.
Yes.
And that's critical, because if that falls apart, the cell doesn't do its function properly.
You brought up the mathematics.
Yeah.
This is so complex. It's just almost really difficult to understand, to comprehend. Could you have a set of rules like underneath that sort of generates all the, a core set or anything here? And is there anything to do with mathematics? How does math fit in?
Sure, yeah. I mean, in some ways, this actually goes back to Turing, Alan Turing, of the Enigma code and the movie recently. So one of the last actually papers that Turing wrote before his premature death was about precisely this question. So what might be simple mathematical rules that could lead to the kinds of spatio-temporal patterns of molecules that one sees inside cells? And he was able to show in this amazing paper, one I would say is, in my opinion, one of the great papers of modern science. He was able to show that two very simple components, one being this process of diffusion or Brownian motion that one actually,
that's right. Einstein's proof of the atom.
Proof of the atom comes from sort of looking at experiments and then doing theory, etc. In 1905. But combining diffusion and simple chemical reactions. So what Turing showed is that if you have chemical reactions, which is like, you mix some chemicals and then you let those combine in various ways and you get some product. Well, normally when you think about chemical reactions, you will the process will go sort of downhill in energy. What that means is that the molecules will recombine until they reach some steady state, and then there's no more chemistry happening. For instance, that will happen in your batteries. You know, while the chemistry is happening in the batteries, you will have your 9-volt battery working. Once the chemistry has done its thing, all the energy has been expended, the battery's dead. So the end result of chemistry usually is a steady state death, in other words. The end result of diffusion is also death, you know, speaking metaphorically, meaning that what it will do is it will equal out the concentrations, the amounts of proteins in different, or any molecules in different parts of the room. So a typical example would be if you opened up a perfume bottle, after a while the smell of the perfume would fill the room, and that's death in the sense that all motion and processes, well, not motion, but the process of spreading would stop. So both of these processes, chemistry and diffusion, eventually lead to sort of everything evening out. And what was absolutely, I would say, an amazing discovery by Turing is that if you combine these two processes, then you can get a pattern, something that's not uniform through space, something that has spatial structure. So you combine two things that have no spatial structure, and you get something, that has spatial, that gets spatial structure. Actually, the sort of the analogy that I really like was brought to my attention by my 16-year-old son, and he pointed out to me that's like shaving cream, because what is shaving cream? It has some fluid, right, and it has some air, and neither of those hold shape. But shaving cream, when you initially when you pour it out in your hand, it beautifully holds shape. So you take two things.
And that's
and
great.
they can't hold
example.
shape, but then you put them together, and all of a sudden you have something that holds shape. That is strange, right?
Yes, it is, but you start to think it's all over nature. But Turing, that's really very unique.
And so Turing was able to show that these two elements, just like the fluid, and the gas that you have in your shaving cream, making something that has structure, he showed that diffusion and chemistry can lead to structure.
Right.
And so that has led us down this path that started in 1953 when this paper was written, which has been to explore these kinds of structures in cells from the points of view of reaction and diffusion. So that's the mathematical language that a lot of us have been using since Turing's times to try to understand what is the fundamental mathematical laws that underlie these
so
these
there is, you are looking for, like, there is that basis. It always seems to be there, doesn't it? Like it's underneath there somewhere, the math, and then the physics, and then the chemistry, and that it's these things just self-organize.
Self-organize, yeah. That's the most impressive thing, right? So it would be, it would be the equivalent of you bringing a bag of bricks and some cement to a building site, and then going for a cup of coffee, and you come back and there's a building. So this is happening all the time inside the cells. These organelles that we were talking about before, they exactly come to being in this way. This way that seems they would be absolutely magical if it were to happen in our world, in the macroscopic world.
Right.
But it is completely possible, and we see it all the time in the microscopic world.
Right.
And that's what makes the microscopic world fascinating. It's like observing an alien technology. It's something that's unfolding in front of our eyes, as long as they're aided by microscopes.
Yes.
But has no real bearing to anything that happens in the macroscopic world, which is, for people like me who get excited about mostly mathematics, fascinating, because then you have to ask yourself the question you asked just a minute ago: what is the mathematics that can actually describe
something is always under there to generate the thing. But partly that was due to the fact that you see these self-organizing systems, and they everywhere, and like ecology all over the place. So even in the macroscopic world, you see this tendency to self-organize, and it's space and form and size all fit in and adjust accordingly. There's something in there that is very widespread, apparently. But in any case, you're into this now. Is this fairly new still?
Well, it's funny. No, it has a long history. In fact, it's one of the first papers that talks about size. Is a paper that was funny enough written by an Englishman whose last name was Gulliver. So that's just
That
perfect,
is
right?
perfect,
I mean, Gulliver talking about size.
But not that one.
But what Mr. or Dr. I'm not sure Gulliver noticed was that red blood cells, which some animal red blood cells do have nuclei, this organelle called a nucleus. Our blood cells do not.
Right.
But many animals do have red blood cells with nuclei. And what Gulliver noticed was that big red blood cells had large nuclei, and red blood cells have small nuclei. And so that was an observation that I think at the time did not lead to further speculations. But then even in the early 1900s, people were noticing things like in developing embryos, what happens is as this embryo develops, in some situations, the cells become smaller and smaller. But as they become smaller and smaller, because they're constantly dividing, but not growing, because they're dividing so quickly, as they get smaller, then the nuclei and all the other organelles inside those small cells become smaller and smaller. So how does that work?
Exactly.
How does the cell know, or how do, since these structures are self-assembling,
exactly.
how are they aware of the fact that they're self-assembling in a smaller compartment? So again, if we were thinking about Lego blocks, the self-assembly is if I scattered a bunch of Lego blocks in this room, and then they, through some magic, which inside the cell is just not magical, it's just diffusion, but through some magic they assembled into a Lego structure. But now imagine I take those Legos and I put them in a smaller room, and somehow they know to assemble into a smaller structure.
To scale.
How do they know? I mean, that's what we're trying to understand.
Right. So that's not fully understood yet. You understand that there are mathematical principles, there are chemical principles, there are, do you know a lot about this now?
Well,
that
we're
part of it, but
we're
the
beginning
scale.
to learn how scale emerges from these fundamental processes. So how is it that something can self-assemble in a way that is cognizant of the size of the compartment within which this self-assembly occurs?
Yeah.
So we have some mathematical models. We were just notified today that one of our papers that describes this mathematical models will be published. But I guess I would say we're still a long ways ahead. I mean, there's still a lot we don't. There's a lot more we don't know that we do know. We know. In other words, what we have now are very specific examples of this kind of scaling.
Yeah.
But we still are searching for very general principles.
Yeah.
And we think they're somehow hidden in these equations that Alan Turing wrote down a while back. But they're so rich that we're still sort of exploring them. And then we're adding new things to them, right? So one of the things that we have to add, it seems, that seems to be playing an important role, is that another mode of transport inside these eukaryotic cells that we show here is not diffusion, but directed transport. So inside cells, there are molecular machines that move the same way the trains move on railroad tracks. The tracks which they move on are filaments that also self-assemble, so that there is filaments that self-assemble inside the cell, and they serve as railroad tracks along which machines literally walk by stepping, tiny microscopic distances, and they use the fuel that's produced by one of these other organelles, namely the mitochondria. And when they walk along these filaments, they can carry with them either organelles or other molecules. And this kind of so-called directed transport, directed because it has a direction, as opposed to diffusion, which is random and has no direction. This directed transport seems to be playing a key role, and this is sort of something that's now going beyond what Alan Turing had speculated at the time, or had discussed mathematically at the time, because it's bringing in a new element and a new mathematical element, because the mathematical description of this directed transport is very different than the mathematical description of diffusion that was really put on firm ground by Einstein, as you mentioned, in 1905.
Yes. This is really fascinating, and you're uncovering a lot of stuff. Does the technology now help you? Because I would think it wasn't available really in Turing's time, was he? And do you use very advanced equipment to get at this, or are you just thinking this still in your heads?
Thinking is still important, and there's no substitute for that, but we're definitely learning a lot by having incredible microscopes these days, which allow us to peer inside the cell and actually watch in real time this
That's
the self-assembly process. So what that really allows us is to take the mathematical models and the theories that we dream up and then test them against reality. I mean, the genius of Turing was that he came up with something that really stood the test of time, even though he had no direct tests of his ideas at the time.
Counterintuitive.
And a lot of his ideas from then have only recently been tested experimentally again, because as you correctly pointed out, those tools did not exist even 20 years ago, let alone 60, 70 years ago when Turing was doing the seminal work.
It's a really fascinating work, though. Very interested in the fact that you go at this with it's multidisciplinary in many respects. Is your team multidisciplinary?
Oh, absolutely.
Okay.
So the way we have physicists, mathematicians, and cell biologists and biochemists all working side by side to really try to crack these questions open. And it cannot, without the mathematics and the physics, it's very hard to make quantitative predictions. It's very hard to get at these fundamental sort of mathematical rules that we talked about earlier. But on the other hand, without the expertise of the biochemists and the cell biologists, we cannot then test our ideas, our mathematical hypotheses against reality. And so there's really no possible way of, I think, moving forward without these different pieces, these different scientific disciplines coming together and working
it's inherently
together.
Interdisciplinary, isn't it? That's a very interesting trend in a lot of areas of science, certainly the interesting areas of science today, that you have to have these multiple minds working together, which must be pretty neat. And on that, I think that at Brandeis you have this very innovative approach to teaching a program. I can remember there were a bunch of letters. Would you mind telling us a bit about it before we close?
Yeah, of course. No, it's something that I'm really excited about. It's really kind of builds on what you were just saying. It's a program that actually introduces interdisciplinary research to first-year students, first-year undergraduates. And that normally is thought to be kind of an insane/crazy idea, because you see, if you want to have students sort of studying something from multiple angles, the thought was always, well, first you have to get them through all of chemistry, all of biology, all of math, all of physics, and that's your undergraduate or even more. That's more that's your master's sort of level type education. Just takes time to master all these things. But then we had a thought, wait a second, I mean, we're getting students from high schools where they've already been exposed to biology, chemistry, math, and physics in their high school curriculum. So why don't we then try to expose them to science the way we practice science as researchers, where those disciplines are all mixed up? They're all come together to answer one question. So what we've done at Brandeis is, with the help of the Hughes Foundation, who gave us a generous grant to start.
Is this the HHMI?
That's the the Howard Hughes Medical Institutes. They awarded us a grant to kickstart this program. And so what we've been doing for the past 5 years, we bring in about 16 to 18 students from all over the country. We sort of have them apply to this special program. And what they do is they spend one semester with us, sort of really kind of learning how to take their high school biology, chemistry, and physics and combine them to look at problems in modern biology. So we'll study, papers, we'll study maybe some classic work like Turing, and all this to kind of get them into a frame of mind where they're sort of deprogrammed, I would say, from what they've learned in high school, and that is that these disciplines have nothing to do with each other. So students come to us and they think physics has nothing to do with biology and chemistry, and it's in own world, and then math is based on all these rules that really have nothing to do with anything with the sciences. So we try to kind of get rid of those biases in the fall, and then kind of the real adventure happens in the spring. And what we've done there is we recruit graduate students and postdoctoral fellows, at Brandeis who work in labs, and they come up with research projects for these undergraduates to do. So under the tutelage of these graduate students and postdocs, they actually go into science labs and Brandeis and start doing interdisciplinary research on these sort of smaller, well-defined projects, which usually build up to a more elaborate sort of real research project that they end up doing over the summer, and then often in their second, third, and fourth years. So the whole idea here is to get these students into cutting-edge research as soon as possible. And what interdisciplinary research also affords is that the boundaries of knowledge here are very, very close to where their knowledge is at. So if you're starting in a discipline, let's say physics, my own discipline, and you want to get to the frontier of knowledge, there's a lot of territory you have to cover. On the other hand, if you're starting, if you're interested in a question of quantitative biology, which is to try to answer, for example, some of the questions you asked earlier, like how do organelles move inside the cell, then the frontier is actually very close by. There isn't much territory to cover simply because a lot of these questions we're asking are very new, in large part because we didn't have the technology to ask them before. And so the students can actually get up to speed very quickly because the amount of ground they have to cover to get to these new, fascinating, cutting-edge research questions is not very large.
Yeah. The other part of that, it seems, it sounds gorgeous. It's a way to learn that's very, very exciting, but it seems to me more natural in a lot of ways, in that a student exposed in this way to science is exposed also to some of the frustrations. You are not going to be successful all the time. This is not graded like that. You have to learn to make mistakes, start over, and feel triumphant once in a while as well. But it sounds fabulous.
Absolutely. I think one of the key goals of this program is to teach students that science is about trying things out, failing, trying again, failing, and then eventually, you get to something new. That is completely sort of absent from the classic way we teach science, where we'll toss a book at the poor student, and then they're asked not to fail. They're asked to reproduce the knowledge that's in those books, and that is completely, as you know, antithetical to science. Science is all about having a dialogue of us humans with nature, and we ask nature one thing, she might respond, she might not respond, and this constant dialogue is really what science is about. It's not a collection of some facts inside a book. That's how we might record things that we've learned along the way as we've held this dialogue. But that's not the business of science. And this is something that is probably the most important thing, that we try to convey through this kind of course.
It's fabulous, and it's the right size. The Brandeis University can offer all of these things, and in a setting probably that's a little more comfortable and so forth. It's fabulous. It was such a pleasure to talk with you, and thank you for all this wonderful information. And I hope we do this again. I want to know more about that undergraduate program. It sounds so great. Thank you very much.
Thank you. Fun.